Go ahead and get started. Good morning, good afternoon, good evening, maybe even for some of you around the world. So we're glad to again have another chance to chat with you. Today we're going to be bringing in a whole cast of characters, many people who care about you, both thinking about the basic science and the clinical aspects. And this is really in response to questions that we've been hearing from you and concerns that you have. And as we're trying to continue to morph what we're doing to address those concerns, I wanted to keep you up to date. And we need that feedback continuously to help answer the questions that are of importance to you. So for those of you who don't know me, I'm Wendy Chung. I'm actually the Chung in the Chung-Jansen syndrome, so that's one and the same. And I've brought some friends with me today. So we've got a pretty packed agenda. We're trying to finish in just an hour, and we may go a little bit over if you've got questions. But I'm going to start by sharing my screen, and I'm going to do a very brief overview. Then I'm going to turn it over to some of my colleagues who are thinking about some of the mental health issues that many people have been talking to us about. And then finally, we're going to do something new, which is going over some of the data from a paper that you may have seen, where we've tried to start functionally assessing some of the variants in PHIP and understanding what they do. And in particular, I know some of you have questions about what that means for your health. And I'll just give you the punchline in case you have to drop off early. I don't think we know yet, but we're trying to think about this with you. So let me very briefly share my screen, and to say that all of this is being recorded and will be posted, and anything that I'm presenting is publicly available, so don't feel like you have to memorize it all. And some of it is in a longer version with similar data that we've previously posted. So I'm going to very briefly just go through the updates. So number one, as many of you have participated in research, I also know that some of you online today are actually new to the diagnosis. And so definitely go back to some of our previous recordings. We all have questions and we're learning together. So I put at the bottom the email contact information for two of our study personnel, Alexa, who's a genetic counselor, and Sean, who's the study coordinator. You can email either of them if you have questions in general, or if you'd like to participate in the research to help us understand this condition better. So in terms of what I'm going to very, very briefly talk about today, we now have about 50 individuals internationally around the world, and we're seeing, as I expected to see, a distribution along the x-axis here of different ages. So this is showing from our little ones, age 1 to 6, all the way to middle age. I'm sure there are more people that are in this bucket here in terms of adults as well as older individuals. They simply haven't been diagnosed yet. And we see approximately the same fraction of males and females. I'm not going to go into detail on the genetics, but we have been collecting your genetic information. And in particular, we've been not surprisingly seeing that this is affecting the brain and behavior. I'm not going to go into this because the psychiatrists are going to talk much more about this. But the point is that not everyone has the same features, and they may change over the life course. And so that's what we're appreciating, and we'll continue working on that. When we think about brain and behavior, we also see issues in terms of fatigue, hypotonia, low muscle tone, hypertonia, high muscle tone, and thankfully it's a small percentage of individuals, but a small fraction with seizures or epilepsy. We do see challenges in terms of cognition or thinking and learning. Individuals continue to improve in terms of learning more information over time. They continue to learn, and all of us are constant learners, so that is something that will continue. It's not degenerative. It's not regressive. We're not seeing that this is a form of dementia. Again, we have fewer adults, but so far, at least in the adults we've seen, we haven't been seeing that. We have, some of you have gotten MRIs or pictures of the brain, and we do see when we can look inside that we see some developmental brain differences in terms of how the brain developed, some anatomical differences, and that may be related to some of the differences in the brain and behavior that we see. We can also see some differences in other parts of the body, although I think some of them are related fundamentally to the brain. Some things in terms of the intestine, constipation, diarrhea, sometimes both, different days. Also issues with the eyes. Many of us, I myself wear glasses, but many of us have some sort of vision issues. Importantly, many of these are corrected or can be addressed by seeing an ophthalmologist. So I want to emphasize that. And then finally, this is the last point that I'll make, is that many of you have heard me say before, there's some other issues, some sleep problems. I think this also can cause problems in terms of behavior. And so one issue I think we'll be thinking about in the future is how to address that. And then also many of you know that there can be issues with weight, and those can be different at different life stages. So in particular, some things can creep up in adolescence and adulthood that we don't see in younger individuals. It's for that reason, and just to give you an update, that PHIP is included in a clinical trial of a medication that helps with weight. This is a medication that's now FDA approved for other genetic conditions, and the medication itself is called setmelanotide. If anyone is interested in this, we do have some individuals with PHIP who have now come through Columbia for evaluation to be in this clinical trial, and some people who are in fact now in the trial. And this is still open to enrollment for anyone who's interested. I'm not going to go into too much today, except to say that all of the expenses are paid as part of this study. So it's something that you don't have to worry about traveling to Columbia. We actually pay for that as part of it, but it is a long-term commitment. And so with this, I realize that may be something that not everyone is interested in, and it is specifically geared for the weight issues. It won't correct all of the issues associated with PHIP. It's meant to address the weight issues and what we call the metabolic issues. For those individuals who do come to Columbia for that weight study, we will also be able to include, in addition, some evaluations that we're doing, in particular with our neuropsychologists and our psychiatrists, to try and understand the brain and behavior better. We'll sort of hopefully help you to get more out of the visit with us. And again, that's all of this is free of charge. That's not something that you pay for. It's part of the research that we're doing. So if you're interested in anything that I've talked about today, again, here are the email contacts for Alexa and Sean, and they'll be glad to either answer questions about any of the studies I've talked about. And if you are interested in participating in research and don't want to come to Columbia or don't have interest in the weight loss, that's perfectly fine. We have options to do things remotely, and you're going to hear more about that from the psychiatrist as well. So let me stop there. And within this, as I said, we're on a bit of a tight schedule. So I'm going to turn it over next to the neurologist, or to the psychiatrist rather. And I think Dr. Jutla, you're going to be presenting. Is that right?
Yes. So let me just go ahead and share my screen here. Hello, everybody.
So while Dr. Jutla is setting that up, I'll say that he is a psychiatrist, a child psychiatrist here at Columbia, who some of you have met, and he's going to talk more about he and Dr. Mule and what they've been doing. And they're really extraordinary individuals who are especially expert in rare genetic conditions affecting the brain and behavior. Okay, Amadee, take it away.
Okay, so I think we had said that I would talk for maybe about 20 minutes. I think it's likely that I will talk for much less than about 20 minutes. So just saying that ahead of time. So I think we may be okay from a schedule standpoint. But basically, I want to talk to everybody about this PHIP related disorder mental health study that we are conducting with Dr. Chung. So I'm Amadee Jutla. I am a research fellow at Columbia, and I am engaging in this study along with my colleague, Dr. Rebecca Muley. She is an assistant professor of psychiatry here at Columbia, and she is here in this meeting as well as a panelist, and we'll be able to sort of make comments or answer questions at the end. So basically, what we know about PHIP-related disorder, about Chung-Jansen syndrome, is that mental health diagnoses and behavioral problems are pretty common in this group, it seems like. So this is based on some of the research that's been done by Dr. Chung and others, and it seems that about 80% of people who have PHIP-related disorder have some sort of mental health diagnosis or behavioral problem. And so these problems include, and Dr. Chung sort of briefly touched on them in her talk just now, they include ADHD, and maybe about half of individuals include autism, and about a third of individuals, but also reported are anxiety, OCD, depression. We're not quite sure what the frequency of those things might be. And along with this, we also have these sort of broader behavioral problems. And behavioral problems are kind of, they're broad in a way where it's not, it's good to know that they're there, but it doesn't help us a whole lot. So for example, aggression is pretty common, but aggression can be caused by a whole lot of different things. And so we know that it's there, but we don't necessarily know what it's coming from, what's driving it, what's the best explanation for it, how we can help with it. Impulsivity is pretty common as well, as is like problematic ingestive behavior. And so we know that these things are pretty common, but there are a lot of unanswered questions. And so we don't know, for example, who's most likely to have one of these problems. We don't know if you have PHIP-related disorder, are you specifically going to be at risk of having, ADHD or autism? If there's a certain factor in play, or is it more random? We don't know if, for example, the problematic ingestive behavior that some people with PHIP-related disorder have, we don't know, does that contribute, for example, to issues with weight that some of those same people may have? It may be related, it may not be related. We don't necessarily know. So there are a lot of just sort of unanswered questions, but we do know that psychiatric problems and behavioral problems are pretty common. So what we want to do is we thought about this as an opportunity to try to learn more about mental health problems in this population. And so we think that if we learn a little bit more about exactly what the problems are in people with PHIP-related disorder, then we're going to know enough to design, for example, future studies of kind of specific disorders. So for example, we could design a study of autism specifically in PHIP-related disorder and kind of look at sort of what's driving that. Or we could look at specifically, OCD in this group and look at what's driving that. We also might be able to kind of develop hypotheses about how does having a variant in PHIP affect brain development? How does it lead to, for example, problematic ingestive behavior? If that contributes to obesity, are there treatments that we could develop for that? That's further down the road, but that's stuff that will be enabled by us knowing a little bit more about some of these psychiatric problems. So with all of that kind of said, I was able to get a little bit of funding to put together a proposal to study this in Chung-Jansen syndrome. And so the study is going to be relatively small. We're hoping to recruit about 30 people. That would be great if we could get 30 people. Later on, it would be even better if we could get more people, but right now the funding is relatively limited, so we're trying to start small. Participation is mostly going to be online because I wanted to think about feasibility. I know that there are a lot of people who are watching this who are in different parts of the country, different parts of the world, and wanted to sort of make sure that we can maximize participation. But that being said, if somebody is, for example, coming to Columbia to participate in another study, we will meet with them and we will do something in person with them. If for some reason, we're scheduled to be, I don't know, at a conference somewhere, and it happens to be timed in a way where we're going to be near somebody in the community, we would be happy to kind of go down there and meet with you in person and sort of try to do the evaluation that way. So it's online participation, but could be in person if and when that's feasible. And we also wanted to sort of make sure that participation would be as broad as possible. I think I'm a child psychiatrist by training, so in the early stages of this, I was thinking, okay, I'll look at children, but actually, there are also adults who have PHIP-related disorder who are on this call and who are interested in what's going on and who have experiences of their own. So I think, it makes sense to have as broad of a participation as possible. So you can participate regardless of your age. And the study, there are really two components to it. So there is one component is sort of more the interactive component, and one component is the sort of more passive component. So the interactive component is going to be a Zoom session or an in-person session, right? Based on what we can do, but probably a Zoom session. And it's going to take it's going to be with me, potentially with Dr. Muley as well, and it will include an interview that will be sort of a structured interview that we psychiatrists like to give, where we ask kind of very specific questions in very specific ways in order to get some information. But there will also be a more conversational part of the interview where we'll ask about your experiences with any mental health problems that you may have had, any behavioral challenges you may have had, and your experiences with the diagnosis, because that's also important to know about sort of what your journey has been like. So we'll do that. We'll also do some more structured things. Like there are a few kind of simple, what we call in the jargon is we call them cognitive tasks, but they're almost like simple games, and they test things like attention, memory. We have an implementation of it where it runs through a web browser, so we'll be able to give you the link, and you'll be able to do the task that way. They're all very brief, and they'll give us just a little bit of a sense of kind of where you're at in terms of your ability to pay attention to things or your ability to remember things, and we'll be able to compare that with norms and compare that with within the PHIP group in order to get a sense of whether people have challenges in that area. So that's the interactive component. The more sort of passive component is a survey that if you're an adult participant, you'll probably do the survey yourself. If the participant is a child, then the interview part is going to be with the child, and the survey is likely going to be filled out by their parents. So this is basically a bunch of kind of rating scales and questions. It will take like at most an hour to fill out, which sounds really onerous. It is probably not going to take an actual hour. I think I kind of try to project that as kind of like the absolute ceiling for like what would be reasonable to for that to take. It should be a little bit less than that, but it may be a little bit tedious, but if we would ask that you just kind of bear with us, because it gives us some useful information as well. And we can use those rating scales in order to get a sense of what's going on. And so we have two ways of looking at things, both from the interview and the tasks and from the survey. So that being said, this is 2 hours of your time. And so we are going to try to reimburse people. And so part of why I wanted to get funding for this small project was to ensure that we would be able to do this. So we'll be able to reimburse people for participating. You'll get a $200 Amazon gift card. So try to make it something that would be, that would be a fair compensation for the time that you're putting into this, so that it doesn't just feel like you're being you're providing us with information and we're saying, it's going to be helpful down the road, but you don't really get anything tangible. So this was important to me to include. And so I'm glad that we were able to include this. So far, we've collected some initial data from a few participants. We've interviewed 3 participants who we were able to get some of this data from. We don't have everything from those participants. And because it's just 3 participants, I would feel a little strange kind of showing you results and saying, here's what we found so far based on like 3 people that we've assessed. So I'm going to hold off on doing that. But as we get more information from people, we'll periodically share aggregate results where we'll be able to say, of these 20 people that we've assessed, this is what seems to be most common. This is what we're seeing. And so we'll be able to do that at these meetings periodically as we collect more data from people. So just wanted to kind of highlight that this is a study that we're doing, that we're excited about, and that we've kind of gotten underway with. You will probably start seeing, we're still trying to straighten a couple of things out on our end, but you'll probably start seeing like an invitation to participate. And I would encourage you to try to participate if you're at all interested. I think it'll be a lot of fun for people to see a little bit more about what's going on, what some of the most common problems are, and I think it'll be really helpful for us to learn a little bit more about what's going on in PHIP related disorders. So just wanted to be able to sort of plug the study, and that's really about all I had. So I think that was substantially less than the 20 minutes that I sort of conservatively said that I might take.
Great. Amandeep, there's a question. Will the study be in various languages or English only? Will it be culturally appropriate for everyone around the world? So, there's some things that might be based on, Christmas versus Jewish holidays and, or Arabic holidays or Muslim holidays. So how does that work?
Yeah, so the study will initially be only in English, which I apologize for, but that's just kind of a limitation based on the kind of the scope of our project. So it's going to be pretty small, to start off with. We just want to get 30 or so people. We're going to expand the scope as we go on, hopefully, but at least to start off with, it'll only be in English. English is unfortunately the only language that I speak. It may be the only language that Dr. Muley speaks. I don't know. She may speak some languages I don't know about, but the instruments that we want to get are all in English. That being said, it will be culturally appropriate, I think, for people around the world. We're not going to refer to sort of specific cultural things as part of the study. Like, that's not something that we're thinking about doing. It's a good and interesting question, and it's something that I'll sort of double check as we're going through and sort of making sure that we're asking things that are universal. But yeah, I don't think that will necessarily be an issue. You can definitely take part in the study if you don't live in the U.S. I saw somebody ask that question. The only limitation is that we're going to be doing it in English is the only problem. And then another question is, would we be able to have another call to review the data sooner than a year from now? That was when our last call was, and to also discuss ways we can help your team acquire funding and support for additional studies for things like how PHIP affects brain development. Yeah, that would be great. I think that I guess it just depends on the speed at which we're able to collect data. But as soon as we have kind of enough that we think it would be worth reviewing, I would be happy to do a call. I think I'll defer to Dr. Chung and her team to in terms of like when we would schedule that or whether we would schedule that. But I think that would be great. And yeah, any ways that you would be able to help us acquire funding would be something would be great to talk about as well. So yeah, I hopefully that answers that question.
Yep. So I'll say we're committed. It's relatively easy for us to set up. It is people's weekend, so I do appreciate that. But otherwise, it's not very expensive to set this up. So I'm happy. I'm going to give us a goal of maybe once we get to 15 individuals that we've looked at as a first signpost to be able to have some more generalizable impressions. And whenever we get to 15, we'll write it down on our calendars. We'll set this up in terms of being able to talk with folks. And then we can continue to move on from there. A couple questions are coming in that are, I think, more genetic related questions. So I'm going to answer a couple of those and then we can go through if there's, I love this discussion. So number one is that as a genetic condition, you only have to test once for this. And if you're negative for it, you're going to continue to be negative for it. And if you're positive for it, you're going to continue to be positive for it. So you don't have to continue testing. This is the one time when you need to do that. We still don't know sort of worldwide what the frequency is, either in terms of birth frequency or how many people out there have the condition. I will tell you that I actually think amongst the relatively new generation of neurogenetic conditions that have been identified in the last 5 years, it's one of the more common ones. And so I don't know if that'll end up being at the end of the day 1 in 30,000 births or something. I'll put that marker out there and we'll see if that ends up being right or not. There is more testing that's being done, and even some work that we're doing at Columbia, trying to make sure that we don't leave anyone behind and identify everyone with this condition from as early as a few weeks of life. We're setting up programs to be able to pilot doing that. And so as we're doing it, I think we'll be able to answer the question and put in places even around the world to eventually get to the point where we can diagnose the condition. So stay tuned for lots of that. In terms of how to enroll in the study, the easiest way is to do the email, and maybe I'll put this back up on the screen for Alexa and Sean's email address and contact information. And then while I'm doing that, Alexa or Amandeep, maybe you can read out another question and go from there.
Okay. Let's see. Somebody asked if our child recently had a neuropsych evaluation, would this be redundant, or should we participate in the study, or should we just send on the report to you? If you recently had a neuropsych evaluation, we would still definitely be interested in you both participating in the study and actually sending on the report to us. That would be really helpful, and that would be great. It would be informative. So that definitely would not disqualify you from participating. Another person asks, is my 23-year-old too old for the study? No. We really wanted to try to have as wide an age range as possible of people included. So.
And just to say, there are a couple questions. We have been seeing many more families where this is passed down in the family. So some of you know that this can, we've got 2 copies of the PHIP gene, and you only need to have a difference in one of the 2. And so sometimes we see it marching down through generations and generations in the family. Sometimes we see it arise for the first time in the individual. So we're seeing both types of patterns. And at the end of the day, we'll see how many fall into each category. But I suspect we'll see quite a few of both.
And I'd just like to pop in and say hello. I'm Dr. Rebecca Muley, and I've been working with Dr. Jutla, who is really spearheading the work on this. So I'd like to compliment Dr. Jutla on getting all of these surveys and tests together. And coming from my perspective as a child psychiatrist that really focuses on neurodevelopmental disorders that are largely attributed to genetic risk factors, the PHIP gene is very interesting in the number of different phenotypes that people develop that have this change. And so I think, hopefully I'm not getting the cart before the horse, but I really feel that this is going to be an iterative process. So as we learn from you in those initial surveys and interviews, we're really trying to get a better sense of what are the main issues that you are dealing with, both as a person carrying that and as family members dealing with it. And I know that one of the things that was brought up previously was the behavioral therapies are also very important. So I think personally, and I think I can speak for Dr. Jutla as well, we'd really like to know about anything you felt was coming up as an issue for you, and as well as anything that you felt was really helpful. And in some cases, medication was not so much the answer, and it was really the behavioral treatments. And that's also very critical for us to know, as certain genetic syndromes, certain types of therapies have been shown to be very helpful. So I'm very excited to be working with you guys and meeting the community. And I'm very pleased to be working with Dr. Chung as well, who's really just a worldwide expert in this. You all know that, but I feel very lucky to be a part of this project.
So there are a few questions coming in about parents getting tested or genetics and it running in the family, and perhaps other questions, not just about parents, but other more extended family members. The best way for me to do this is to deal with this one-off, so to speak, because each family has their own issues. If you do email Alexa in particular, that's the email address she put up, which is ag4216@cumc.columbia.edu. If you email Alexa, we'll help in terms of arranging genetic testing through other family members, and we'll kind of cascade it or ripple it through the family. I just want to emphasize what you just heard, which is that I really truly think you guys have the answers immediately. I think you have the answers within yourselves in the community in terms of understanding whether they're, I call them life hacks, but what are things that you figured out that have worked? What are therapies, educational strategies, ways of getting through the day for fatigue, medications? Like, I think you've got a lot of information in your own lived experience. And I think if we can aggregate this together, and the reason we're doing this with more than just the online surveys we've done in the past is I'm convinced there's some richness or some detail that we're going to have to pull out of you to a certain extent. And so that's what we're doing with this next phase of things, is because I think by having that discussion, we're going to learn together much faster. And I know you've done a lot of that online in the Facebook group and just individual chats with each other. And so we're going to try and do this in a more coordinated way. And what I hope comes out of this, just to be very explicit, is that I don't know the timing, but I hope we'll be able to come up with some standard of care guidelines. And so what that means to me is that we'll be able to say, it'll be based on our impression. I will admit it's going to be not quite the evidence review that we do for some things, but it'll be our best guess, at least for what we think should be done in terms of caring for you and your family members. And that, in terms of I always ask, what's in it for me? Like, why should I care? That's actually really important, because then when you go to get services, when you go to have these services paid for, when you try and advocate in your schools or at your job for certain issues related to disabilities or related to specific ways to support you, by pointing to such a document and saying it was published and it's a medical recommendation, your local doctors can actually advocate for you better. Your educational providers, your behavioral providers, all of them can do that. So it ends up, I've seen it being really powerful for you. And so if we can work together to get this done in the sooner the better, I think we'll be able to, like I said, make a real influence for you. And that's short of anything in terms of what you're going to hear about with the science and where I hope someday we can get to in terms of the science and doing magical things with Gene. I think these are very concrete next steps that we can take together as a community. Okay. So there's a question here. My wife has Ehlers-Danlos syndrome, and a couple of other parents we talked to have children with this syndrome and Ehlers-Danlos as well. Have we found any links between parents who have different syndromes like this and having children with the syndrome? So this comes in a sort of complicated way, in the sense that I have heard from many of you that you have, I'll call them fatigue and connective tissue disorder types of problems. I do think some of those are a little more frequent with PHIP, and Ehlers-Danlos syndrome in many cases is a clinical diagnosis. It's not something that we can read out and document in the DNA. And I think for some of you, if you're seeing the PHIP and those connective tissue issues or those fatigue issues only coincident, that is, they're only in the person with PHIP, I believe it's probably not a second diagnosis. It's simply a manifestation of the PHIP. Now, separately, I know there's some of you where there really is a second diagnosis, and there may be other family members without PHIP who have some of those connective tissue disorder problems. And that can be true as well, because connective tissue problems are fairly frequent in the population, 1 to 2% of people that have them. And so it could just be true and unrelated. That is, you're allowed to have another condition that's running in your family, and it doesn't have to be part of PHIP. So I know that was a little bit complicated, but I think this is what we're trying to figure out. And by having someday 100 is a magical number for me. If we can get to 100 people, I think we can be closer to being confident of what we're putting in clinical care guidelines. And do we need to say anything specific about connective tissue problems? I hope we'll have enough depth of data to be able to get at that. Okay. Alexa, I know you've been monitoring the questions and shuffling them around. Have we gotten everyone who's out there so far?
Yep, I think we are caught up on our questions.
Okay, very good. So we'll go to the next segment, because I know some of you have been interested in this. So the next segment, we have a very special guest with us here in the United States. Dr. Jin Grewala is a basic scientist, and he's been studying this gene actually for quite a long time. And so he and I have been thinking about different genetic variants in the gene and whether or not it has other effects beyond just the brain and behavior. And he studies things, as he's going to talk to you about, more biochemically. And so he doesn't study people and brains and behavior in the same way that the psychiatrists do, but I would say equally important are his contributions. So I'm going to let him share his screen, and then we'll have time for questions at the end.
All right, thank you. Betty, can you all hear me?
We can hear you. It's a little fuzzy, but we can hear you.
Oh, it's a little fuzzy.
Go ahead and keep talking.
Can you see my screen?
We can see your screen.
Okay. I'll try to fix the audio. Hopefully this goes to what it gets problematic, but please do let me know.
It's not great. We can hear you, but it's kind of soft and it's a little fuzzy.
Give me 1 second. Is this better?
That's better, much better.
Okay. Yeah, so I'm not sure what's wrong with the settings. I'm speaking through my microphone, and when I do that, I cannot really hear you all. So I'll have to keep switching between the audio and the mic. But for now, I'll just go ahead and cover my presentation. So let me start off by thanking Dr. Chung for the invitation as well to be able to present my findings today. Thank you all for participating as well. It's been really insightful learning all about PHIP related disorder in altogether a different angle that pretty much I don't really think about on a daily basis, right? So anyways, just to provide some context, I am Huzaifa Dungrawala. I'm a faculty member in the biology department at the University of South Florida. And my main, I would say, the goal of my lab is to perform basic biology and try to understand the inner workings of life processes. And the process that we are most interested in is DNA replication. And so just to highlight, the main goal of my lab is to really understand how components contribute to the duplication of the genome. So here is just one example that I call it as a replication fork, which is quite well known in the field as well. And so when we think of replication, these are really the sites where replication is performed on DNA. And to do this job, we have many proteins that come together. These are the actual machines that help make sure DNA is duplicated. So I won't go into much of the details in terms of the background, but I just want to provide some context. If you think of DNA replication, it's by far a highly conserved mechanism across all life systems on our planet. And since we must have seen this before as well in our very basic biology curriculum as well, where if you have a cell and it has 2 copies of a given gene, and which is often the case for every other gene as well, that DNA is duplicated, and then eventually after cell division, those copies are transferred to the two daughter cells. And this, of course, happens across generations. And I think so this is what Wendy earlier mentioned as well, that we have 2 copies, and then oftentimes we have either one or both the copies being mutated. So in the context of replication, one thing to bear in mind is that although it may sound very trivial, DNA is by far the most fragile in this situation because it can be easily damaged. And when DNA is damaged, it can cause mutations in the DNA. And when they are carried forward, or if they're problematic, that results in a variety of human diseases as well as conditions. So in order to deal with this damage, the cells rely on an entire pathway to deal with the DNA damage events. And there are many proteins that are involved in contributing to this function. And one such protein that we work on is DCAF14. Now DCAF14 is the protein name for the gene PHIP, correct? So when we think of PHIP, it's the gene annotation, and the protein product that really functions in the cells is called as DCAF14. So we, about 3 years back, we stumbled across DCAF14, and what we identified in a publication earlier in 2021, when we published this report, we show that in cells that do not have DCAF14, they have problems replicating their DNA. And this is especially in contexts when you expose the cells to DNA damage conditions. And so consequentially, what happens in these cells is they're unable to maintain replication, and over time they also build up unstable genomes. Okay, so this is what we published back in 2021, and sometime just last year as well, Wendy and I got in touch, and we really wanted to find if the observations that we see in our laboratory could also perhaps be extended to variants that are found in PHIP, correct? And one idea behind this question was that there are many diseases or human conditions that are linked directly to replication, and so hence we got in touch, and shown over on the bottom over here is a kind of a map where we were able to isolate various variants in PHIP that were known to exist. And so from this map, what we wanted to pursue and ask is to pick 2 variants, and highlighted our 2 variants over here, the D488V and the E963G, and ask, when these variants are expressed in cells, do they show the same phenotypes that we observe in cells that do not have DCAF14, right? And so if we see these conditions mimicking the phenotypes, then perhaps we can also extend these phenotypes to individuals as well that have the PHIP related disorder. So I won't go into much of the details in terms of the experiments, but we can rely on two common assays and techniques that we can use in the lab to study these consequences at the level of replication. And so shown on the left is just a readout for replication assay, where we can track replication at single molecule level. And in this graph, if you see a distribution with higher values, means that physiologically that's close to what one would expect in sort of a wild type condition. And anything that's at the bottom is where we now see that there are defects during replication. And so this is what we see is that when we express either the two variants of PHIP, you have problems replicating the DNA. And on the right is what we see a technique where we can assess genome stability at individual cell level. So of course, bear in mind, this is all looking at individual cells, right? It's not looking at organism level. And on the right, what we can see is there is increased genome instability as well, again, in situations where we are expressing both these variants. And so I'm sure most of you are aware of this publication that just came out about a month ago. And we were able to at least directly link the functions of decaf14 in replication directly to the mutations or the variants that are found in PHIP. So what does this really mean in this broadest sense? This table is very busy. I don't mean to go over this in detail, but it's a really nice review from one of the really well-established leaders in our field, Dr. Simprich. And in 2014, her review really characterized and gave a glimpse of how defects in replication can be directly associated with human diseases. And so shown over here in this table, listing all the human diseases that are known to result from defects or problems in replication. And so there are many such proteins that you can see. Some of these are the Fanconi anemia pathway, the Bloom proteins, which is known to cause premature aging and growth retardation as well, and plentiful more. And I think so the take-home message over here is that every protein is distinct, right? It performs its own function in order to recognize what is happening at the DNA level in terms of replication. And so, of course, when you look at the characteristics as well, they're pretty diverse. The spectrum is quite broad. And when we compare these characteristics to PHIP-related disorder, we see a few commonalities, but not at a major scale. So one commonality we see is dysformisms. And then if you look at the characteristics that are slightly different, we can see that many of these conditions have growth retardation as a common characteristic, as well as increased predisposition to cancer because of mutations that do occur in the DNA itself. So to wrap up, what are we considering moving forward? Well, just to highlight, our findings are looking at these variants in a very specific cancer cell line, because we are a lab that looks at replication and its consequences in diseases. We pick cancer as a model system. But of course, what's important for us is to see if we can recapitulate or perhaps see these exact replication phenotypes in a cell type and more so a tissue specific type context, right? And further, it's important to even identify additional mutations that are present worldwide. As we have seen before in a presentation, we see this rather a distributed spectrum of variants on PHIP. And so it's important to further nail down what additional mutations are present. And then eventually really nail down what decaf14 does. I think Amandeep reflected on this earlier, right? It's important to know what the protein does in order to do its job. And this is where we are highly invested as a basic science laboratory. We have 3 projects trying to further understand and expand on our understanding of what decaf14 does during replication. And that really gives us an added advantage to then ask, how do the variants affect these functions of TCAF14. And so with that, I think so this is the last slide, and I'll be happy to take any questions.
Great, that was fantastic. I know there are some questions already. I'll say that for the Cold Spring Harbor case report paper, Alexa, I'm going to tag you with getting that to Elise and Joyce, a copy of that. And then also, I just want to reassure people that there's a lot that we don't know. I think that's one of the messages. We're very early in our understanding of this particular protein. And although you were seeing a lot of other diseases associated with other factors, other genes and other conditions, it is not as if I think this condition, the PHIP related disorder, is going to have all of those other features. And I know some of the questions I've been getting that I don't yet know the answers to are what are those features that we might see in particular in adults. And so cancer, I'll let you riff off this, but cancer is one of the questions that families and people are concerned about. Again, I want you to remember the distribution of what I showed you earlier, that most of the individuals we have in the community are still children and young children. And when we think about cancer, it's not just one particular genetic variant that leads to cancer. It's oftentimes the accumulation of multiple changes over a life course that leads to cancer. And so we may not, big if there is cancer associated with this, we might not be seeing things until the 40s, 50s, or 60s. And so that's part of at least my sort of inclination to try and look in the families that we've identified where this is inherited, to look at some of the older generations and out to other adults to see if we're seeing any enrichment for cancers. But Huzwaifa, I'll let you comment and riff on yourself in terms of knowing that this is just, hypothetical. This is just pontificating. This is not based on data.
Yes, let me switch over again.
Yeah, so just to reiterate, what Dr. Chung just mentioned, this is very hypothetical, very initial observations as well. As I've mentioned, many of our research is really geared towards linking towards some disease aspect, some conditional aspect, right? And as I showed you in the table, at which we have extensively had conversations with Wendy before as well, that there's a massive distribution in terms of the characteristics that are commonly found in other diseases that are more associated with replication, as well as this increasing disposition of cancer. And so, of course, this is very early stages, and it's a big if in the sense that whether time disorders would be impacting developing cancer at this stage, right? And so this is something that we all should keep in mind when we think about considering what are the effects of it.
Okay. Unfortunately, as you're switching back on that audio, it's really hard to hear. But I think the take home is that unfortunately we don't have as many answers, and it's conjecture. But again, I think we know where to focus some of the attention in terms of issues. I'll say that some of you have clinical questions, and Alexa, I'm going to ask you to put our phone number in the chat. So if you have a very specific clinical question for your own child, yourself, whoever it is, we're not going to discuss those in a group. We're going to keep this at a high-level conversation across individuals, but you can either email Alexa or you can call that phone number if you want to make an appointment for a clinical evaluation. Another point that people are making, and this was one point that I wanted to make about the study that we had done, is that most individuals in the community, I went by this very quickly, but have what we call a mutation or a genetic difference in the gene, which we know, or we at least are pretty certain that it's going to destroy or take out one of the two copies of the gene. And we can make pretty good guesses about that. There's some other individuals in the community that have what we call missense variants. It's that one single amino acid substitution. And one of the reasons we did the experiments we did is that we weren't sure whether or not how it affected the protein or if it affected the protein. And for some of you, you're in what I call purgatory, in the sense that you may be the only person in the world known to date state that has the particular variant, that missense variant in particular, and we're not sure whether they all act the same or they all affect the function of the gene. My fantasy personally is that we would have a scalable assay. The experiments that you showed, I'll just say, number one, were extremely laborious, very expensive, very time consuming to do. And right now they're not scalable so that we could do it for everyone that we wanted to. That's my fantasy about what we can do someday, but we're not there yet. That is really important to me because for all of those different genetic variants, I don't know whether everyone's within PHIP is going to act the same way, and I'm not sure they're all actually the answer. And so there's still some more work we need to do. For those of you where that particular genetic variant is new in your child, and where those features your child has seem to fit the pattern that we're talking about, there's a pretty good chance that's your answer. And again, we can talk with you offline, or you can email Alexa and we can go into deeper conversations. Some of you have been reading or asking about, what about gene therapy? I've heard about CRISPR, I've heard about gene editing, I've heard of these things. Is that going to be an answer? And what I'll say to that in general is that in the short term, that's not going to be an answer. I'm not saying in the long term, there might not be possibilities, but in the short term, we really need to aggregate our information for educational strategies, medicines, other things that we can use that are already available to be able to, in the short term, for the next few years, be able to get to where we need to be. In the long term, I will say that there are other things that are coming up in terms of within companies or within universities. And I do have optimism that in the longer term, there may be different strategies than what are available today. But we've got to think about what I call the care until the cure. So we've got to think about the care in the short term until we can get to the cure. I will say that we can try and, for those of you who are in Florida and thinking about whether or not you can stop by the University of South Florida, just to say that don't come there to get a medical opinion, but I'll let you decide whether or not you want to see anyone who might want to visit your lab.
They're more than welcome, absolutely, yes, but there's no clinical opinion on my side, but I can definitely explain the science.
Okay, very good.
Yeah.
Okay, someone is asking about, there may be other families where there's cancer that runs in your family, or there may be another gene for cancer that runs in your family. And we talked about other genes for Ehlers-Danlos. There may be a CHEK2 or another gene for cancer in your family. Again, we don't know the answer, but I do have concern that if there's already sort of a fire, if you will, would this add gasoline, or would this add fuel to the fire? It's possible. I don't know, but those are the types of things we're wondering and thinking about. And as my goal would be that longer term, if we can come up with the answers to think about whether or not we need to do enhanced screening for cancer, if there is something. But again, that's a big if. I will say someone asked a question earlier that they were surprised that DCAF14 isn't lethal, or, why don't we see a problem everywhere in the body? Why does it seem to be tissue specific? Do you want to hazard a guess?
Yeah, I'm not, I think that has, can you hear me, Wendy?
We can, yep, this is good.
Okay, yeah, this is great. Yes, so that's a very interesting question. When you think of the kind of the model system or the kind of cell line we are working with, I was surprised to see that when we created these knockouts in our cell line, that they were just fine, that they were not, they just grew without any defects in proliferation. So I guess this cell line specificity, and this also could attribute to the fact that there are many protein products of the gene PHIP itself, right? When you think of DCAF14, that's your full length protein product that PHIP gene produces. But then you also have other protein or other isoforms of PHIP, such as NDP1 is there. There's another version that's a truncated version of PHIP as well. And so, of course, we are just focusing on one such protein product of PHIP, right? And so maybe that could explain why you do not see these lethal effects across cells or even the entire organism.
Okay, very good. So there's another question. I think I answered this, but just to make sure, where your particular family's variant isn't listed in the databases that you may have access to. So there are things called ClinVar and mutation databases and other things. And that is possible. It's possible that it's only been seen once and that the laboratory that did that testing hasn't uploaded it to any of those databases. I will say that there's oftentimes a lag of up to a year, sometimes even more, between when a lab sees it and uploads it to the databases. So it may be coming soon, but it's not there yet. For any of you that didn't know your mutations were being uploaded to databases, just to be aware, it's not with your information. No one knows who you are, but it's simply in the service to make sure other people can get diagnoses in terms of those. Just to say that anything from today in terms of the recording specifically will be given to the family group leaders so that they can post this on the website, just like we've done with every other presentation we've done, so that you can share it with your doctors, other family members, or people who weren't able to be here today. I'll also want to put a shout out. I don't know if I know some of you are new to the group. Elise and Joyce are two, I think of them as Wonder Women. So they're two mothers who are out there in Europe and in the United States who have done a fantastic job in terms of keeping on top of things. It's a real labor of love in terms of keeping the group organized, keeping the website organized, helping in terms of coordination for today's event. And they help me in terms of being able to hear from you. So when they're hearing or seeing things that I might not be aware of, they're always the first to come to me and raise my awareness so that we can make sure your voices are heard. So I just want to put a shout out to the two of them. They don't get paid for doing this. They literally just do this as a labor of love, and they're fantastic at doing it. I'll also say, and then I know we're going to need to close if no other answers come in, or no other questions rather come in, or that we really are very dedicated to this particular condition. I have to say, I didn't name it after myself. I'm not anyway that egotistical, but once it did get named after me, though, I have to say I have a real sense of responsibility. So for you guys, you got lucky that someone came up with the idea to name it Chung-Jansen syndrome, and so we're with this. We're with you for the long haul. We are doing this as unfunded research largely for doing this, and to the extent that it's not going as quickly as you might want it to do, I will say that's because we're all doing this really as a labor of love to you guys in the community. I think over time, as we're hearing from you about what are the key questions and understanding and putting together some of the basic science with the clinical questions and trying to come up with better answers in the future, I do think we're going to need to do this in a more organized way. We're just sort of in our infancy, almost embryonic in terms of both identifying you guys out there and for us developing the questions and understanding the lived experience. And so I can feel this sort of inflection point, though, and I'll say this being a doctor who works with a lot of rare diseases, and I really do feel like it's more common than many of the other conditions that we see out there. And I think there are a lot of generalizable things that are important, like I said, for a lot of other people besides just you guys. So people that have to do with diabetes and weight issues, with behavioral challenges, with potentially maybe things like cancer in the future. I don't know, we'll see. But I hope in terms of doing that, we'll learn some things that are not helpful just literally to you all, but to like some of you that don't have PHIP and your family members as we're going through. So with that, I do think there are lessons learned that more people are going to be interested in, just to not make this all about rare diseases, but also thinking about common things that many other people are struggling with. Okay, so with that, we're right at the top of the hour. I want to be respectful, especially of the doctors and scientists here who have donated their weekend time. If there are questions that you didn't feel comfortable posting publicly, like I said, feel free to email Alexa or Sean. If they don't know the answers, they'll direct them around to the other members of the team that you've seen here. And do sign up in terms of any of the studies at Columbia or any of the online studies. And if you have particular ideas or observations that you think are interesting and important for us to be thinking about, do please individually contact us. And I just realized I myself forgot to put my email address up, but if you Google me, you'll find it in 2 seconds. So feel free to email me directly, or Sean or Alexa will direct traffic. So with that, we'll end for today, but I'm looking forward to hopefully getting together online at least in maybe 6 months or so, whenever we hit that 50 number, or 15 number rather. And then I'll just put it out there. I would love to actually meet some of you face to face. So if you all think about at some point having an in-person meeting, wherever that might be, Hopefully we're coming out of COVID and there are ways of being able to travel and get together in person, but I'll just plant the seed. If you guys decide you want to do that, count me in. Would love to meet with you guys. So with that, I'll wrap up. And again, we'll have the recording posted shortly. Thanks, everyone. Bye-bye.